Spotlight, lectures and performances on and around Albany State University.
So far, she secured many big grants from NIH and wrote more than 50 peer-reviewed journal papers. So I am very fortunate to have her as my mentor in my own breast cancer research, and Dr. Schoenlein's institution, that is Medical College of Georgia, is our mentor institution. Let's welcome Dr. Patricia Schoenlein to today's presentation.
Can you hear me?
Yes, ma'am, I can.
Oh, okay. So, Dr. Saha, thank you very much. But the truth is I'm a really small fish in a big sea, but I want to be a big fish. So, what I wanted to talk today about is autophagy. And I'm just curious how many of you could define autophagy for me? Just raise your hand. Okay, if I ask a different question, how many of you could define apoptosis for me? Anyone? Aha, now see, there are 4 or 5 hands that are raised. So I'm not going to ask you to define apoptosis. I just want to tell you that when I get about halfway through this talk and you want to go to sleep and you're feeling a little bit bored, that autophagy is as important as apoptosis. And one of the reasons that we have not made more progress in studying certain diseases is because we've been misled. We have been studying solely apoptosis for about 20 years. 5 years ago, if you would run a Medline on autophagy, you would pull up maybe 30 papers. Maybe 7 years ago, 30 papers. Now if you type that word in, you'll pull up thousands and thousands. Because we realize that autophagy is as important as apoptosis. And that's why I wanted to put it in this talk today, because I want you to understand a little bit about it. But the talk is about breast cancer. I'm sure that all of you know that breast cancer is diagnosed just in the United States in about 200,000 women a year. And we have very good treatments for a lot of the breast cancers. But we're in 2012 and there is surely no cure, and there is surely no good treatment for certain types of breast cancer. So let's just think about breast cancer for a little bit. So you can do your own palpation in the shower, try to check and see if you have a lump. And a lot of women detect breast cancer that way. It's the same for men, right? You examine your prostate, see if there are any lumps or your testicles. So often we diagnose our own breast cancer. Alternatively, if we have health insurance, we typically will get a mammogram once a year. And, you know, I was thinking maybe a pointer would be good. What do you guys use? This? I got it. I can do it with this. So this is a mammogram here. And this area right here is where a tumor is. So if you go and you get a mammogram, thank you, then you know that you have a tumor. So what happens at that point? Well, the breast cancer patient has now been told that there's a tumor and there needs to be a biopsy. So there will be a biopsy. It'll be sent to a pathologist. And that pathologist is going to determine whether that breast cancer is estrogen receptor positive or not. If it's estrogen receptor positive, we can do a targeted therapy. So I want to go over estrogen receptor a little bit with you. This is just outlining for you that you have your core biopsy, it goes to a pathologist. pathologist determines whether you're ER positive. Typically, if you're estrogen receptor positive, you'll be progesterone receptor positive. Now, there are women who are also HER2 overexpressing, and those women are not necessarily estrogen receptor positive. However, this HER2 is a receptor, and those women can also have a targeted therapy. So, of the 200,000 cases a year, 70% are estrogen receptor positive. The other 30% are estrogen receptor negative. Estrogen receptor negative breast cancer patients I won't talk about today, but they're going to be put on chemotherapy. So that's after the surgery, the removal of the tumor, maybe the removal of the breasts, radiation treatment for local control. They're going to be put on a cocktail of drugs where you have 5 or 6 different drugs in the cocktail. They're going to lose their hair, they're going to be sick all the time, and they're going to pray that that tumor regresses, all that metastatic disease. That's if you're ER negative. But if you're ER positive, you're kind of hopeful because you know you may not have to be put on chemotherapy. There are treatments for estrogen receptor positive breast cancer. So, first of all, the estrogen receptor, the pathologist does an immunocytochemistry stain. In other words, there are antibodies that will ID these cells. They look brown compared to this ER negative breast cancer. And that's telling the pathologist that that female has a cancer that's expressing this receptor, estrogen receptor. That's good. That female's happy because she probably will not go on chemotherapy. So what is she going to go on? She's going to go on an anti-estrogen therapy. So let's look at that for a minute. First of all, the receptor, the estrogen receptor that I talked about, that's only 1 in 30,000 proteins in our cells. But the fact is, it's expressed at high levels. Whenever you have a protein that's at high levels in a particular type of cancer, you can target that protein. You try to block its action. So estrogen receptor positive means that that patient will have targeted therapy. So let's just look at the estrogen receptor. It's a protein. It's actually not on the cell membrane. So if this is your cell, this is your nucleus, this is your estrogen receptor, ER. And this estrogen receptor will bind the estrogen that's in female bodies. And that estrogen, when it's bound to the estrogen receptor, drives proliferation of breast cancer in those 70% of women. So that estrogen receptor, it can be in the cytoplasm, it can be in the nucleus. When estrogen binds it, then estrogen receptor acts like a transcription factor. That means that it goes and it sits on the promoter of genes and it drives the transcription of those genes. So actually, if you're estrogen receptor positive, the estrogen receptor is bound by estrogen, that's going to transcribe about 100 genes in that cell. So if you would block that estrogen receptor, you could block the transcription of those genes. So all of you, I'm sure by now, have studied the cell cycle, right? You could probably draw the cell cycle for me, right? Gap phase, S phase, G2, M. If you go through the cell cycle, you lead to 2 cells. 1 cell becomes 2. That's what happens in your cancer cells that are proliferating. The cells are replicating, but no cells are dying. Now, if you can block the estrogen receptor in those 70% of breast cancer patients, what happens is the cell cycle stops right here at G1S. So there is no proliferation. You're halted at G1S in the cell cycle. All right, now I want to back up for a minute. Can we block the estrogen receptor? Yes, we've been doing it for 25 years. We have an agent called tamoxifen. Tamoxifen was identified by Craig Jordan. It was part of his PhD. Now he is truly a big fish. He's one of the biggest fishes. This is a renowned scientist, and as his PhD, he ID'd tamoxifen. And he showed that this compound could bind the estrogen receptor. And when it binds the estrogen receptor, it prevents estrogen from binding. So estrogen cannot drive the transcription of those genes. That leads to growth arrest. Now, tamoxifen is called a selective estrogen receptor modifier. You'll see that term SERM. If you're diagnosed with breast cancer and you start Googling because you want to get really smart about your disease, you'll see that term SERM. That's what it means, and in a minute I'm going to define it a little bit further. Okay, so anti-estrogens that are called SERMs are like tamoxifen. That means that in breast tissue, they will block the growth-promoting activity of the estrogen receptor. But on the other hand, in bone and in our neurons in our brain, it acts like an agonist. It acts like estrogen. So actually, as a patient, you can be put on tamoxifen. You can block the growth of your breast cancer, but at the same time, you're not losing all your bone density. You're not developing osteoporosis because this is a selective estrogen receptor modifier. So its action is tissue specific. So it's been a very, very good drug for a very long time for estrogen receptor positive breast cancer. All right, now this is a little complicated, but I'm sure some of you have already had biochemistry, some of you maybe not. But when we talk about estrogen receptor alpha being a transcription factor, that means that it binds to a particular place in the promoter. Now that's after it's bound by the ligand estrogen, and it'll drive the transcription of many genes, right? That's what that means. And I just want to mention to you now, because I have a slide coming up, that it's not that simple. It's not just estrogen binding the estrogen receptor, and that's all it is. There are these other proteins that we refer to as co-activators and co-repressors. So you have a complex that's sitting at this promoter. So for estrogen to act like an agonist, these co-activator proteins are recruited to help drive the transcription. And for tamoxifen to act as an antagonist in the breast tissue, there are co-repressors that are recruited to that promoter area. So now we're talking about a complex that's causing the estrogen receptor to have a particular function. And that complex involves other proteins. Now, we called tamoxifen an estrogen receptor modifier. Tamoxifen is not the only drug in that family. There are other drugs here, like raloxifene. And raloxifene actually was developed to prevent women from getting osteoporosis. These women were put on raloxifene for 5 years. Osteoporosis was prevented. So when you see Sally Fields on TV and she looks great at the age of 60 and her bone density is really good, it's because she's taking raloxifene or something like it. And what they noticed in those women is in that cohort of individuals who had the raloxifene compared to the ones who didn't, they had a much lower incidence of breast cancer. So actually, raloxifene is a SERM, but it wasn't even developed to treat breast cancer. It was developed to prevent those of us who go through menopause and lose all of our estrogen from losing our bone density. But now it is used to treat breast cancer. Now, you might say, why do we need raloxifene? You just told me tamoxifen is so great. Very shortly, I'm going to show you that a problem in the clinic when you treat women with estrogen receptor, so the women are estrogen receptor positive, 70% of the 200,000 that come in, they will be put on an anti-estrogen like tamoxifen. Of those 70%, 30% will not respond well. So it is not like if you are estrogen receptor positive, you are cancer-free. You don't know if you're going to be one of the 30% of women who don't respond. Because of that, they started looking for other drugs. That's why they brought raloxifene into the clinic. If I'm an estrogen receptor positive breast cancer patient and I put on tamoxifen and I fail, my cancer's not, regressing, and that clinician sees that, in a couple of months he's going to switch me and he'll put me on another SERM like raloxifene. And it has turned out that if you switch from one SERM to another for a while, you can keep those women who aren't responding in kind of a homeostatic situation. But typically, if the woman is estrogen receptor positive and she doesn't respond well, you can only switch those drugs so much, and the cancer will come back to haunt and possibly kill the female. Or the male, because males also get breast cancer, but it's 1% or less. Okay, so why is that? So I want you to think about this. We talked about tamoxifen. I showed you the cell cycle. I told you that what tamoxifen does is it causes growth arrest. Well, what is growth arrest? Growth arrest is like you want to kill somebody and you say, okay, I'm going to starve you to death. And so it's going to take, 30 days if all you give them is water, maybe 6 weeks, right, to die. That's a slow death, right? That's what growth arrest is like. It's a very slow process. So you've got tamoxifen, you give the woman tamoxifen, she takes her pill every day for 5 years. The studies were done, and the study said you have to give it at least 5 years. Because if you only give it 2 or 3 years, her cancer is going to come back. But if you give it 5 years, there's a chance it won't come back. The reason for that is because the primary action of tamoxifen is cytostatic. It causes growth arrest. That's very different than chemotherapy. Chemotherapy, when you put a patient on chemotherapy, it kills cells. It doesn't often growth arrest them. It will kill the cells by apoptosis. So that would be like, okay, I'm going to kill you, but I'm going to shoot you with my gun. And then you die the same day if you shoot in the right place, right? But the problem with chemotherapy is there's no targeted therapy. You give the patient chemotherapy. That could be, there's a whole slew of drugs. Probably the most popular one for breast cancer is Taxol. Taxol comes from a plant that some poor non-funded researcher identified and isolated the Taxol. Okay, literally, he was not funded. And it has turned out to be one of the best chemotherapeutic agents we have. What is the target of Taxol? The target is microtubules that are in the cell. But the problem is that the Taxol is going to affect everything, right? It's not selectively going after, say, estrogen receptor positive cells. So that means it's going to kill all the cells that line our GI system. Well, that's why we throw up all the time when we're on chemotherapy. And you know, you can only regurgitate so long until you have to lower the dose or stop. That's why our hair falls out. That's why we get mouth sores. I mean, chemotherapy is very toxic. So you can only get to a certain dose. And that's it. The patient can have no more. Their bone marrow can't tolerate it. So you'll kill a lot of the cancer cells, but you often don't kill all of them. It's not a targeted therapy. You have to have a differential between your normal and your cancer cells. So with tamoxifen, it is a targeted therapy. And for some women, it works really well. But it is a cytostatic therapy. It causes growth arrest, not death. Death would be a cytotoxic therapy. So for 25 years, people have been trying to figure out, in the women that don't respond to tamoxifen or to some other SERM, what can we do? Can we profile the women when they come in the clinic so we know which ones won't respond, and we'll give them tamoxifen and chemotherapy? That's one question. The other question is, well, why is it they're not responding? If we could identify the mechanisms of resistance, maybe we could target those and the women would respond better. All right, so this is just a little scheme showing you that tamoxifen as a first therapy up until 2 years ago was the most common therapy. If you failed, then you'd be put on a second-line therapy. That could be like raloxifene. And if you fail again, you'll be put on something different and then something different, and chemo will be added. So why do these cells resist the tamoxifen? Why do some women not respond well? So for 25 years, we've been studying mechanisms of resistance. And here it is in a nutshell. There are co-activators that are amplified. Remember I showed you that graph? And I said you need co-activators and co-repressors at that transcriptional complex. Well, those can change in resistant cells. For instance, there's this one protein called AIB1, which is a co-activator, and it can be present at very high copies in certain breast cancer cells. And if it is, that will give you tamoxifen resistance. You would say to yourself, well, maybe there are mutations in that receptor. Maybe that's what happens. You give them tamoxifen and the receptor mutates, and so that's why they're resistant. Well, that's very uncommon. Very, very uncommon. That is not a common mechanism of resistance. So co-activator changes, co-repressor changes. And then there are these two guys, PI3K and MAPK. Now, those guys are kinases that are part of a signaling transduction cascade, and they can be upregulated in cancer cells. We know that they can be, so the last 5 years have been dedicated to making specific small molecules that will block these two kinases. And so you let the female fail her therapy, fail the tamoxifen or the raloxifene, and once she's failed the therapy, you say, okay, I'm going to now give this PI3K AKT inhibitor. And the reason I'm bringing that up is I heard Dr. O'Regan give a talk about 4 weeks ago. So she's a medical oncologist at Emory. And actually, there were women who failed first-line anti-estrogen therapy. They gave them AKT inhibitors, and there was some regression. So it was a very exciting study from that perspective, because it was the first example of targeting a kinase. A kinase is a protein that phosphorylates other proteins that had some type of outcome that was positive. So what that's telling you is that for the last 15 years, all the money that's been spent to target kinases has been ineffective. Now we have a study that's effective, so that's a good thing. But it's been a very slow process. Now, we showed that one mechanism of drug resistance could involve the process of autophagy. And autophagy, I'm going to define it for you just so that you're familiar with it, because if you stay in school or you go into the medical profession, you're going to hear more and more about this biological process. So I want to just go over it with you. And I just want to tell you my thinking. That doesn't mean it's accurate thinking. There are other people who think like I do. That doesn't mean I'm going to pull a big NIH grant in. But it is what I truly believe. I don't believe that you start out with the cancer, you treat the woman or the man, and actually the 1% of breast cancers in men are usually very aggressive, and they can be estrogen receptor positive. But you start out with the cancer, you put them on a therapy, an anti-estrogen therapy, and you know that 30% are going to fail, but you go ahead and they fail, and then you turn around and say, okay, I'm going to enroll you in a clinical trial. I can give you AKT inhibitor, I can give you MEK inhibitor, I can give you this, that, or the other. I don't think that's the best strategy. That's the strategy we use, and it may be the only strategy we can really get approved in America. But I don't think it's the best strategy, because initially you have a tumor that's heterogeneous. And if you let it continue to progress to the point where the resistance mechanisms are expressed, meaning that you have upregulation of that AKT we talked about, or this other kinase, or upregulation of the epidermal growth factor receptor, at that point that cancer cell is really smart. I give it the name genomic instability. What that means is that the chromosomes in that cancer cell are very unstable. And you can give it one drug, and yeah, maybe you can kill 90% of the population. But this is not like CFTR. This isn't like cystic fibrosis transmembrane regulator defects. In order to treat cancer, you've got to really kill every cell. If you leave one cell behind, that one cell could mutate, right? So if you're at the point where you're already able to upregulate and overexpress these resistant mechanisms, you're past the point of what we call good cell cycle checkpoints. You're in the phase of what we call genomic instability or mutation. And when you're at that point, what the doctor does for you is he keeps you, I like to use the term maintenance therapy. Actually, that term is used now for colon cancer. Colon cancer is so hard to treat, and it's a terrible cancer. And if you have colon cancer, you'll get your colon resected and you'll go in, and really what your medical oncologist does is he keeps you on maintenance therapy. If you had metastatic disease, that means every 6 months he changes your chemotherapy. So he's not curing you, he's just trying to kill the cells that develop mutations that come out by switching drugs. So I think we don't want to be at that point. We want to figure out why it is up front that the cells that get the anti-estrogen therapy are surviving. Once we know that there's what they're doing, how they're surviving, we could give the anti-estrogen therapy and target the survival mechanism. One approach to that, which I don't study, but Dr. Jan and Dr. Sal's collaborator at MCG does, are called stem cells. I don't know if you've ever heard of cancer stem cells, but cancer stem cells could be the part of the population that's very resistant to therapy, and that's being investigated. But another mechanism that we were able to identify is autophagy. What we found is that as soon as tamoxifen or any SERM is used to treat breast cancer cells, yes, they go into growth arrest like we talked about, but they do more than that. They start a process of autophagy. And when you're in autophagy, you're very resistant to hormonal therapy and to other forms of chemotherapy. So what is autophagy? So autophagy is a stress response. Our normal cells can do it, our cancer cells can do it. So most normal cells have a basal level of autophagy. Autophagy means to self-eat. Self-eat, that's what it means. Now, in cancer cells, you can induce autophagy to high levels. So if you treat with Taxol, etoposide, those chemotherapeutic agents, or you treat with hormonal therapy, the autophagy is upregulated. All right, well, let's look at other things that induce autophagy. Hypoxia induces autophagy, so that would be lack of a blood supply, areas of the tumor that don't have blood. Starvation induces autophagy. Reactive oxygen species induces autophagy. So autophagy is a process that occurs in the cytoplasm of cells. So this is your nucleus. What's that? Nucleolus, that's where ribosomal RNA is made. This is your cytoplasm. So this is a cell. This is an electron microscopic representation of a cell. Here's your nucleus in this cell, and now there are all these little circles. And these circles are called autophagosomes. So if I just had a schematic, what I would tell you is that when a cell feels stressed, all of a sudden 28 genes are expressed in that cell, and a little organelle is made called an autophagosome. How many of you know what a mitochondria is? Everybody. That's an organelle with a membrane, right? So is an autophagosome. In fact, an autophagosome has a double membrane, just like the mitochondrial membrane. But the difference is it's made to higher levels under stress conditions, and it has a specific role. So if I'm starving a cell to death, autophagy is induced. When that double membrane forms, there are constituents in the cell that get trapped, like the mitochondria and proteins. And then that autophagosome with these cytoplasmic constituents fuses with the lysosome, which all of you probably know is the hydrolytic machinery of the cell. That's called an autolysosome. And all the stuff inside gets degraded into the building blocks. Those building blocks get spit back out into the cell, and the cell will only make the most essential things that it needs to survive. The analogy I could give you is if you're being starved to death. What does your body do? It starts to break down your fat stores, right? Well, this is kind of like that. Like, I'm starving to death, and I need key nutrients, so I'm going to recycle everything else to give me the key nutrients. You know, the problem is when you're in this autophagic mode, when this has been induced, what happens is the cell doesn't die due to the starvation. It has figured out a way to overcome the starvation. And so instead of dying right away, you have 6 weeks. Well, during that 6 weeks in a cancer cell that's undergoing autophagy, mutations can develop, and the cell can come out of this mode and proliferate. So autophagy is a regulated stress response that has only been defined fully in the last 5 years. Now, you can look at autophagy a lot of different ways. We have all sorts of research tools to look at autophagy. And now in cancer patients, you can look at tissue to see if autophagy is going on. And there was a recent paper showing that colon cancer cells from patients had very high levels of autophagy, higher than normal tissue. This is from one of publications, and I know this is hard to see, but this is your nuclear envelope, and this cell is packed with autophagosomes. And if you can't see that on this EM micrograph, this dye is a specific dye called monodansyl cadaverine. Every blue dot you see there is an autophagosome. Under normal conditions, you don't see autophagosomes in this cell line. But we exposed it to anti-estrogen therapy over time, just like a patient. And this is what happened. Autophagosomes built up at super high levels. And needless to say, the cells were not even growth arrested by the hormonal therapy. See all of these pretty dots? These are normal cells, and all of that punctate staining there are the autophagosomes that are induced by chemotherapy and anti-estrogen therapy, if you look at the literature. So we knew that autophagy was building up in the cells. In a minute, I'll show you that if we block autophagy while the cells are getting anti-estrogen therapy, we can kill the cells. So I have this little pedestal here. You have ER-positive breast cancer cells, and you can either survive after growth arrest if you have autophagy, or some of the cells might die, but not many. But the balance here is being regulated by autophagy. So what we've added to the picture is hormonal therapy like tamoxifen. Yes, it induces growth arrest, like many, many researchers have shown in the past, excellent researchers. But then there's another stage called macroautophagy. And in order to effectively treat ER-positive breast cancer patients, you must block autophagy. Now see, anytime you have a regulated process, and I told you there are 28 genes, that means you can target the process. So we will be able to block autophagy. And look at the difference. This is an anti-estrogen resistant cell, and when you treat it with this inhibitor of autophagy, look at the difference in cell number. It's a tremendous decrease, and an increase in death. Compare that amount of signal to that amount of signal. Since we've ID'd autophagy as a survival pathway, there have now been 5 other reports that agree with us, even though it took us 2 years to get that one paper published. Because at the time, people didn't look at autophagy as a survival pathway. So now other investigators have confirmed that induction of autophagy is pro-survival for breast cancer cells undergoing hormonal therapy and other types of chemotherapy. So why not block it here before you get to that point? Well, that's a key question, because how are you going to block it? You need an agent to block it, and you need the agent to get into the cells. This agent here, chloroquine, how many of you are familiar with chloroquine? Because it treats malaria, doesn't it? Right? In third world countries, in Africa, we have used chloroquine to treat patients who get malaria. So malaria is caused by a parasite. And the reason it works is the organism that causes malaria, it goes into the autophagosome to hide. And when you add chloroquine, chloroquine literally blocks the autophagosome lysosomal turnover. So we've been treating malaria for 25 years with chloroquine, not even realizing that the way it's working is by blocking and getting rid of the hiding place for the parasite. But now that we know that, we actually have chloroquine in clinical trials to treat certain cancers that we think are going into autophagy. That's the one agent that we have that might work. There's a caveat I won't go into today as to why it may not work, which means that we may need better agents. So there is an approach, and we think that we do need to block autophagy early, and we are doing our animal studies now with chloroquine plus tamoxifen therapy. So in the last 5 minutes, I'm just going to finish up a couple of things. So all I've done is introduce the concept of anti-estrogen resistance, multiple mechanisms that it can happen, but if you target too late, the cancer cells are so dynamic that everything you throw at them, they're going to mutate to. So we're saying, why not target the autophagy up front, not after the patient relapses, but when you're actually giving the tamoxifen therapy. That's what we've talked about. Now I want to just tell you a little bit more about anti-estrogen therapy. A few years ago, aromatase inhibitors came into the clinic. So what we've talked about is using a SERM like tamoxifen that actually binds the estrogen receptor. But now if you go to the doctor, if you're ER positive, you'll probably only be put on the SERM if you're in a small rural community and can't get to a good medical health center, or if you are premenopausal. In other words, you haven't gone through menopause. Because menopause that happens between 45 and 55 years of age, it's defined by your ovaries shutting down. So in the female, most of our estrogen in our system is made by the ovaries. Just like in the male, most of the testosterone is made in the testes. But the man, for some reason, gets to have his testes active forever. That's not true for women, right? We're only given a certain period of time and that's it, ovaries shut down. Well, you'd think that that would solve the problem. But I said most of the estrogen comes from the ovaries, not all of it. In a postmenopausal woman, you can have estrogen in your system. And the reason for that is your adrenal gland, which sits right on top of your kidney, it has a zone of cells in it, and those cells make androstenedione. That's strange because that's a precursor to testosterone. But we do, we make it, and we secrete it into our bloodstream. Our blood has aromatase. That's an enzyme. The aromatase will convert the androstenedione to estrogen. So you see, a postmenopausal woman takes precursors of male hormones that are made in our ovary and turns them into estrogen through the action of aromatase. 30 years ago, there was a female who said, we need to block aromatase in postmenopausal patients. And if we don't, that little bit of estrogen will drive breast cancer. Everybody thought she was crazy, but guess what? 25 years after her first publication, industrial companies have made aromatase inhibitors. You've heard of letrozole. That's an aromatase inhibitor, and that's different than a SERM. An aromatase inhibitor is an inhibitor that will block that enzyme aromatase. So if you give that to a postmenopausal woman, she will have no estrogen. That means she's going to have osteoporosis early, right? Because there is nothing to support her bone density. And anything and everything you can think about that would come with estrogen depletion, terrible hot flashes, etc. So I'm not saying it's a perfect drug, but it is better. So this is a very telling study. When we put 70% of 200,000 patients a year on tamoxifen, our good response rate, that would be partial response and complete response, is right at 20%. Do you realize that that means all those other women will relapse or maybe even be resistant initially? So when they compared that to Femara, which is a type of aromatase inhibitor, you see you have an extra 10% response. Well, that doesn't look that great, right? You'd like to see something up here. But it's good enough. It's an improvement such that most medical oncologists will put you on an aromatase inhibitor as a first-line therapy. However, there is still resistance to aromatase inhibitors. And we've done work in our lab, and we know that aromatase inhibitors lead to autophagy. So because it's, I'm going to finish up my seminar by just telling you a couple of things. I won't even go over that because we've probably done enough. So the bottom line is this. If you're diagnosed with estrogen receptor positive breast cancer, you thank God that you're estrogen receptor positive because you know your prognosis is better. That doesn't mean that you're going to respond well, and it doesn't mean that you're going to respond long term. So that therapy in and of itself has to be improved. Now, if you want to wait until the female develops metastatic disease, that's an approach, but that's a very late approach. So we need really good researchers to think about the different cancers and to think critically and creatively in how we can find new targets to make therapies better. The best we have is estrogen receptor positive breast cancer treatment is on a pedestal. That's probably one of the better cancers you can have. If you're estrogen receptor negative, your prognosis is very poor. And if you're triple negative, it's almost a death sentence within a certain number of years, aside from prayer, which I do believe in. Colon cancer, pancreatic cancer, all these cancers. I mean, we have been funded for 40 years, and we have made progress, but do you see it's very slow? Cancer cells are very smart. So for any of you who are interested in research, you have 3 or 4 people that I collaborate with here, Dr. Jan, Dr. Sao, Dr. Saha, that are all looking at different aspects of breast cancer, or go on and get your advanced degree. I know the funding is tight now, but honestly, we need young minds thinking about the disease relative to their demographics, you know, to improve this, because this isn't just a female problem. I mean, there are kids that are left without mothers, husbands without wives, you know, wives without husbands, and that's just for this one cancer. So we're starting to profile, right, to figure out what everything is, but just good basic research, dissecting something like autophagy is really key. And I'll conclude by telling you this. These are the people in my lab who did the work on the 3 papers we've published on autophagy. As we were doing our work, it became crystal clear, and if you run a Medline on autophagy, you'll see this. You know, the Alzheimer's disease we've been trying to solve, that's also a terrible disease. My dad had Alzheimer's. For years, we thought it was apoptosis killing all those cells. That's probably not it at all. It's probably defective autophagy. So autophagy is involved in neurodegenerative disorders, in stroke, in cardiac problems, in cancer. It's a very, very critical biological process that needs to be unraveled. And that's it. I hope I didn't bore you. Thank you.